Okay, so what we're going to be looking at then is HIV, human immunodeficiency virus. You need to be able to know what the structure of it is and how it acts as a pathogen. This is the last little bit in our cells bit of section 2, and it's the bit that links in a little bit to everything else that we've covered already. So one of the things that we've already covered and that you should be able to do is draw and label a virus. So the section that talks about prokaryotic cell structure and viruses, that's where it comes from. I'd like you to have a go now. I'm going to give you a couple of minutes just to draw a virus and then have a go at labelling the different bits. Okay, hopefully you've got almost all the way through that. We are going to look at the structure of HIV in particular, which is an example of a virus, and hopefully that will remind you and you can check yours. So, HIV, what is the structure of HIV and how does it act as a pathogen? You need to be able to describe the structure of it, how it behaves as a pathogen, explain how it replicates in quite a lot of detail, and then finally, if you're aiming for an A at the end of this, you need to do a bit of research about treatments of HIV. So the specification says you need to know the structure of the human immunodeficiency virus, HIV, and you need to know how it replicates in helper T cells. What you should be thinking here is, oh, helper T cells, lymphocytes, remember that from the lesson, and hopefully we'll remind ourselves that that's a cell-mediated response, and hopefully we're thinking, oh, replication in one of those, that's not going to be good. So we'll have a look a little bit at that, and that's how it behaves as a pathogen. You also then need to know how HIV causes the symptoms of AIDS. AIDS is a product of HIV, if you like. It's a consequence of it. So someone can be HIV positive and they may never develop AIDS, but if they're left untreated, then they will develop AIDS, which is quite serious. And why antibiotics are ineffective against viruses. So specifically looking at a virus now, HIV being the example we're looking at. But we'll look at generically why antibiotics can't be used. So a little bit of background then. HIV stands for human immunodeficiency virus. If we look at these words, we break them down. We've got human. It's only in humans. You cannot contract this from another animal, and you cannot, if you're a human with it, pass it on to another animal. It's immuno, so it's going to be all based on the immune system and how the immune system works, and it's a deficiency, so it's going to inactivate or suppress the immune system. It's going to cause it to be defective, and it's a virus, so we should be thinking all those things from when we looked at the structure of cells and viruses. It's acellular, it's non-living, and it doesn't have the same structures as any cell. So it causes the disease AIDS, and AIDS stands for acquired immune deficiency syndrome. So it's something that is acquired, it's over time. So someone can become HIV positive. That doesn't mean they're definitely going to get AIDS, but it could result in AIDS if it's left untreated. In terms of contagious diseases, this is a relatively new one. The first case was diagnosed in 1981, so that really is very recent. It's contagious in that it can pass from human to human, host to host, but it needs quite a lot of contact in order for that to happen. So HIV will be passed on through bodily fluids, and that's got to be more than just sweat or saliva. So this is through sexual intercourse or injecting of drugs. Those are the biggest ways that this spreads. So it can affect certain people in the world who do not have access to condoms and contraceptives, or people who are addicted to drugs that they inject. So heroin addicts, for example, can be quite a pocket of the community that have HIV, are HIV positive. So the structure of HIV then, this should be ringing some bells in terms of the structure of a prokaryotic cell. Surrounding the HIV virus, we have this envelope okay, which is made of lipids. It's called the lipid envelope, and it almost acts as a little bit of a casing, okay? I'm not going to talk about it being like a cell membrane because it's a very, very different structure, but it is this idea of something surrounding the virus as a whole. You can see we've got these attachment proteins, and they are embedded within that lipid envelope. They are going to be a specific shape, just like all proteins are in their tertiary structure, and they are going to be able to trigger responses and also bind to things. Once we're inside then, if we look inside the virus, we've got the matrix, which is just another layer inside the lipid envelope. And within that, we've got this more rigid structure called the capsid. And the capsid, inside it, contains the genetic material and enzymes. The genetic material that is in viruses is RNA, not DNA. So this is ribonucleic acid, not deoxyribonucleic acid. And the enzymes present, among others, are reverse transcriptase. Okay? Now, transcriptase is the enzyme that is responsible for putting or turning DNA in a nucleus into RNA. And actually, reverse transcriptase does the opposite of that. So it's going to take RNA and turn it into DNA. And that's a really important key point when we look at how HIV acts as a pathogen. So, as I've said, we've got the lipid envelope, which surrounds the outside of the virus. We've got attachment proteins embedded in that lipid envelope. We've got the capsid, which is a protein layer inside the envelope. RNA, sorry, typo, ribonucleic acid, which is the genetic information of the virus. Reverse transcriptase, which is an enzyme catalyzing the reaction of RNA to DNA. And then a retrovirus is another keyword. It's not part of the structure. It's a keyword. HIV is an example. A retrovirus is a virus that can change RNA into DNA. So HIV is a good example of that. So how does HIV actually act as a pathogen? How does it cause problems for the host? So it's a virus, which means that it doesn't replicate on its own. So it has to get into host cells in order to replicate. It needs the setup of a host cell in order to be able to do that. So the first thing is then it enters the body. And when it enters the body, it's going to go straight into the bloodstream. And once there, it's going to circulate around the body. Remembering that our bloodstream is there to get nutrients and to get toxins away from every single cell in our body. So once a pathogen gets into that bloodstream, then it can get everywhere in the body. Once it's in, what it does is it binds to proteins, okay, called CD4 proteins, and you do need to know that name there. So a protein on the HIV virus, so one of the proteins that are embedded on the envelope, will bind to a protein called CD4. And that CD4 protein is found on surfaces of membranes, especially on helper T cells. So HIV will now bind to the helper T cells. Once that happens, the protein capsid fuses with the cell surface membrane. And once that has happened, that opens it up, almost like a vesicle fusing. It's going to fuse and it opens it up. So the RNA and the enzymes of the virus are going to enter the cell. So if we just flick back quickly to the structure, this is the capsid here. And if that then fuses to the cell membrane of the helper T cell, all of the material in here, so all the RNA and all the reverse transcriptase, is going to enter the host cell, the helper T cell. The HIV is then going to turn within host, this is meant to say within host, I do apologise. HIV turns RNA into DNA, and that's then going to go into the nucleus. So now the virus, which can only contain in itself RNA, has managed to turn the RNA into DNA using its reverse transcriptase and all the things available in the host cell. And now that DNA, which codes for that RNA, is going to now enter the nucleus. Once in there, it's going to undergo the first bit of processing for protein synthesis. So mRNA is going to be formed, and that's going to enter the new HIV cells. Also, the mRNA will make proteins inside the host cell. So using the ribosomes of the helper T cell, it's going to come out of that envelope, the nuclear envelope, it's going to come out of the nucleus, it's going to find a ribosome, and it's going to replicate and produce proteins. Once we've got these proteins being made, we've got HIV particles now made. Now those can go and spread more around the body. The HIV virus itself then, the original one, is then going to break away from the helper T cell, and rather than just drifting off down the bloodstream to find another helper T cell, it leaves the helper T cell surrounded in a lipid membrane. So all the manipulation of that RNA and DNA has now resulted in more HIV particles being produced and this lipid membrane. So what that does is severely hinders the T cell. So HIV, as I said earlier, someone can be HIV positive. That doesn't mean they're going to get AIDS, but left untreated, HIV will cause AIDS symptoms. So the way it does that is it interferes or kills the normal functioning helper T cells. And what we should be thinking here is going back to those lymphocyte lessons. Helper T cells are required for that cell-mediated immunity, that cell-mediated response. So as soon as helper T cells are triggered, they then produce cytotoxic T cells, B cells, phagocytes, and memory cells. If we do not have enough helper T cells, or the helper T cells are just inadequate, they're not going to function, then we don't get all those consequences afterwards. So without enough T cells, one of the biggest problems is then the host, would not be able to produce enough B cells. And if there are not enough B cells, they're not going to be able to produce enough antibodies. So contagious diseases and pathogens that have antigens are not going to be able to be responded to effectively. Equally, if there are no cytotoxic T cells being produced, then the body of somebody who is HIV positive may well develop cancerous cells, but the body's immune system would not be able to deal with that. So the body becomes unable to respond sufficiently to infection from external non-self tissues, but also cancerous cells and self tissues that are out of control. So HIV itself is not a direct killer. Somebody could become HIV positive and have it living in them, and the HIV virus itself would not cause them to die. However, what it does do is prevent the immune system from functioning, and that will cause ill health. It could encourage cancer to develop because nothing is controlling those out of control cells, and eventually, therefore, that can lead to death. So, on a bit of a sideline then, why don't we treat HIV with antibiotics? Very same reason we don't treat a cold with antibiotics or the flu with antibiotics. Antibiotics are no use against viruses at all. An antibiotic is a chemical compound which is going to go into the body and interfere with the metabolic pathways of bacterial cells. Metabolic pathways are any pathways that involve a series of chemical reactions, so respiration, for example. If those bacterial cells then are living in a host body and antibiotics come in, the antibiotics are going to interfere with those metabolic pathways and stop them from working. They also can interfere with the bacterial cell, which is made up of the protein called murine, and that's going to allow water to enter by osmosis, and then the cell will burst. A cell wall is there to keep a rigid structure. Without the cell wall and the rigid structure, then there'll be serious problems. So we end up with osmosis happening here, and then cells actually burst. And obviously if we burst a cell, that cell is now dead. Viruses, however, are not cells. They are acellular. Therefore, they do not perform their own metabolic activities. They cannot do respiration. They cannot do protein synthesis. That's why they have to get into a host cell in order to survive and to replicate. So if there is no metabolic activity, then antibiotics have nothing to interfere with. They are chemicals that interfere with metabolic processes. If they cannot do that because the virus doesn't have a metabolic activity, then it's not going to have any effect at all on the virus. Also, something to remember is that viruses go into the host cells, so they don't just sit on the surface, they go into cells, and therefore there's no mechanism for antibiotics to attack. In a bacterial cell, it can attack the cell wall, it can prevent metabolic processes, but in a virus that just doesn't work. So, I don't know if any of you have seen these in doctor's surgeries, for example. Antibiotics do not cure colds, do not cure flu, do not cure viral infections. What they can do, and occasionally they can be given, is if somebody's suffered with the flu and they have developed a